Okay, so today we are going to talk about cell communication. And cell communication is fairly complex, but we're going to break it down pretty simply into just the major general points that happen anytime cells communicate, and then break into a few examples of different ways that cells communicate with one another. So, here, as always, is a breakdown of what I expect you to know by the end of this lecture, with some practice, of course, from class. But you should know the three stages of cell communication. So first there's reception, then there's what we call transduction, and then there's response. And then you need to know how G protein coupled, so coupled with the G protein, receptors receive signals and start transduction. So that's going to be the part of the reception phase. Okay. How receptors tyrosine kinase receive cell signals and start transduction. So I really just want you to understand two different ways that transduction may begin. Okay, that's really what these 2 points are getting at. I don't need you to memorize these crazy terms. Okay. I want you to know how a cell signal gets amplified by what we call a phosphorylation cascade. So phosphorylation is not a new term for you. Okay, we've seen that. It's anytime you add a phosphate group. And we're going to see that here too. So we're going to break down what a phosphorylation cascade is. And then how a cell response in the nucleus will turn on genes while in the cytoplasm it activates the enzyme. So if you have a cell responding, the cell response may occur in the nucleus, in which case it's going to affect the genes because that's where the genes are, or it can affect the cytoplasm, and therefore it will affect the enzymes directly. Okay, so there's some comparing and some contrasting you're going to have to do there. And then finally, you need to know what apoptosis means and why it's so important for normal multicellular organisms like ourselves. Okay. So we're going to watch this in class, but in case you're curious, you want to watch it again separately, anything like that, there's this interesting TED Talk on how bacteria communicate with one another. And so we are going to watch that, and we're going to discuss how that is similar to and different from multicellular organisms. But we're going to jump right now to animal cells. Okay. Now, animal cells are going to communicate with each other through a number of different ways. There's direct contact, and that's through what we call gap junctions. Okay, so a gap is, of course, like a hole, if you will, or like a space between them, and a junction is where two things meet. Okay, so a gap junction is going to be a place where two cells meet, but there's still some movement between them, right? So there's some holes that stuff can get through, and we'll talk about that. And then there can also be the secretion of local regulators. So these are things that regulate something in the cell or in a different cell that the cell will release, or the body may release, and then that'll interact with those cells. Okay. And then we can also communicate long distances between cells using hormones. Okay. Now, your book talks about a whole bunch of different signaling types. It talks about local signaling and long distance signaling. And I do want you to know that hormones are for long distance signaling. Okay. But I don't need you to memorize paracrine signaling or even synaptic signaling. So these are all really useful visuals for how this works, really useful visuals for these different forms of cell communication and signaling, but I don't need you to memorize every nitty-gritty detail. I want you to realize what's happening here and what's happening here and why it might be happening. So why is this neuron putting some stuff out, and what is it going to do to this neuron? I just kind of want you thinking about that. And then why is a hormone going to travel in the bloodstream? Okay. Now, anytime you have cells communicating with one another and signaling to one another, we have three stages. I broke these down earlier, but we have the reception stage. Okay, so this is where the signal molecule, whatever you sent, is actually going to come towards the cell. Okay, so it's coming from outside the cell. So if I'm a cell and my neighbor is a cell, he's going to send me some kind of a message, maybe it's a text, and communicate with me. So that message, that molecule that they're sending, is a ligand. Okay? After that has been received, we entered the transduction phase. During transduction, we take that signal that we received, so that text, and then we actually convert it into a form that we can do something with. So something is going to happen inside of the cell, inside of my house, that's going to take whatever information they told me, and I can now actually physically change something. So maybe I left my water running outside in the front yard, and my neighbor sent me a text and said, hey, just so you know, your water is running. And now I've received that information. So that is the reception phase. I've received the information. And then I have to get up and I have to go towards the door, right? So I am now taking that information and I'm going to start actively using it. And then when I actually go and turn off the water, that would be the response. So this is what the cell does in response to the signal. Okay, so this is the overall solution, if you will. So I was told the water was still on. I had forgotten about it, so I got up and I went and turned off the water. That's my response. I turned off the water so I'm not wasting water. Okay, so reception can look a number of different ways, but here's a nice simple example. So here we have extracellular fluid, so this is outside of the cell. Here we have the cytoplasm, so this is of course inside the cell, and therefore this yellow part, or orange I guess, is our plasma membrane. Okay, so that is our cell membrane, our phospholipid bilayer. Okay, now here we have a protein. This is a protein that spans across the entire cell membrane, and it has a spot for this signaling molecule. So it's going to receive it. So this is a protein that we call a receptor because it will receive something and it will detect, hey, there's a signaling molecule here. Then during transduction, so here is where this reception phase was where I was getting the text. Okay, your cell got the text. And then here we have transduction. So during transduction, this protein will send that message in some way throughout the cell. And this may take multiple intermediates. So it may go through multiple proteins or multiple organelles, depending on what the signal is. But it's basically just transforming that message into a more usable form. Okay? And then finally, you have the response, whatever that response is. So that response can be changing your gene regulation. It can be, it could honestly even be the cell committing suicide, which is what we call apoptosis. Okay, so it can be a lot of things, but for now, I just want you to know this general response, okay? This general, I should say, signal pathway. Okay, so this is how we generally receive signals and then eventually respond to them. So let's talk about the reception more. So typically, there's going to be that signal molecule, that's that ligand, and then the receptor. Okay, so this is our signaling molecule, also known as our ligand right here. Okay, so I could write ligand or ligand. It really depends on who you ask. Okay, that's right here. And then that's going to bind to our receptor. Okay, this is our receptor. And that's a very specific relationship. Only this ligand should be able to fit here because you don't want this thing going off when the ligand isn't there. Okay. So that's what we mean when we say it's highly specific. Now there's multiple types of receptors. There's plasma membrane receptors. So these guys, excuse me, are going to be part of the plasma membrane essentially, and that's kind of what we've seen. But there's also intracellular receptors. So sometimes the ligands can actually make it through the cell, okay, make it into the cell. So sometimes the ligand doesn't have to go here to a protein. Instead, it might be able to find a way through here, right? Remember, anything that gets through this plasma membrane is typically going to be small. It's going to be hydrophobic. It's not going to be polar or anything like that, okay? And then some examples of that, just in case you're curious, are testosterone and nitric oxide. Okay? And then basically anytime a ligand binds to something, that's going to cause the protein to change its shape. And anytime you change a shape, you're changing what it's doing. Okay? Because form leads to function. So if I change the form, I change the function. So that's going to cause our transduction signal, our transduction pathway to begin. Now, in the plasma membrane, receptors category, there are a few different things that you'll see pretty regularly. Okay, you'll see this thing called a G protein coupled receptor. You'll see tyrosine kinase. You'll see ligand-gated ion channels. This is all really fancy ways of representing this right here. Okay, so don't get too bogged down in it. That's my recommendation to you. Don't let it freak you out, because it's just more complicated versions of this. Okay? So, with the G protein coupled receptor, basically, the book talks all about this, and it talks about how there's all these different G proteins that will interact with different things. I don't want you to get bogged down in it. Okay? It's more complicated than it needs to be. This is the actual process. Here's our G protein coupled receptor. It's in our plasma membrane. There's a G protein down here, but it's inactive right now, and then there's an enzyme. Basically, something's going to bind onto the receptor. That's going to cause the G protein to activate, which is then going to activate the enzyme. So it's just like a middleman. You don't need to memorize it, but I do want you to understand that there are multiple ways that this can happen. So plasma membrane receptors are the receptors that are in the plasma membrane, just as a reminder. And here's a little summary table of what I just described in that G protein coupled receptor. You don't need to memorize it, but it would be useful to you if you remembered that the G protein plus that GTP, that's the thing inside of the cell, is actually what activates the enzyme. Okay? Here's the second one. There are some similarities here in that we have a ligand, and that ligand is going to bind right here. Okay? And then that's going to cause a change inside of the cell, and that's really all I need you to know. It's going to cause a change inside of the cell, and that's going to lead to our cellular response. Okay? So I'm going to skip over this. You don't need it. Here's our final example. So a ligand will once again bind, and that will cause a gate to open. And this gate is literally just a channel. So it's just going to let some stuff through. In this case, it's going to let some ion, whatever this green ion is, through. And now it can get into the cell, and now it's going to lead to some kind of cellular response. Again, don't memorize it. It's just a different, fancy way of this happening. Okay? Those three things I broke down are all this, except three different ways. Okay? So with that, let me skip over. All right. So that was all reception. So let's move on to transduction. So in transduction, we have what we call cascades of molecular interactions. So cascading, I want you to kind of think as like the domino effect. One thing happens, and that causes another thing to happen, and that causes another thing to happen. Okay? So there will be one molecular interaction, and that will cause another molecular interaction, and that will cause another one, another one, another one, until you eventually hit the target molecules. So whatever it is that you want to happen, okay? So one enzyme that your textbook will talk a lot about is protein kinase, and this is an enzyme that will actually phosphorylate and activate proteins, okay? So this is actually going to be responsible for kind of the response side of things. So you should just be familiar with this enzyme. I don't need you to be able to draw it or anything like that. And then there's phosphorylation cascades. So this is basically the same idea as the domino effect, except specifically with adding phosphate groups. Okay? So here's what it might look like in a real cell. So we have a signal being received here. We have some kind of transduction, and then that will eventually lead to a phosphorylation cascade. Okay, so this is all part of transduction. And then that will eventually reach our target molecule, our active protein, and then that will eventually lead to our cellular response. Okay? Don't memorize it. Just realize that, hey, this takes energy, and that should make sense because you're doing a lot of things. So there's ATP being used up. Okay? Now, there can be these things called second messengers, or sometimes they're called secondary messengers. And these are going to be small molecules or ions that can also send a signal. Okay, so they can continue the signal. I don't need you to memorize any of these examples, but your book likes to talk about the C-amp and the calcium ions a bit. Don't memorize it. Okay, I want to move on to response. So the response phase is our third phase of cell communication. And this is going to be where you actually regulate the protein synthesis by turning your genes on and off if you're in the nucleus. Okay. So, or you can regulate the activity of proteins in the cytoplasm. So what I'm going to show you first is the response if it's happening in the nucleus. So here I have reception, then transduction, and then eventually my response will reach in the nucleus. Okay, so that's where I am here. And here I have my DNA. And normally you don't want to mess with DNA, right? Because DNA is eventually going to get translated into mRNA, and we're going to talk all about this when we get to gene expression. But if you change this up a little bit, you can change what proteins you're making. So your DNA is responsible for coding for the protein. So like your DNA is telling your body, make this protein, okay? Or it's giving instructions on how to make a protein. But sometimes you don't want that protein. Sometimes you only want that protein if you need it. And so this allows us to basically control how much of that protein we're making at any given time. So this is one form of gene regulation. This is where we regulate our gene expression. Okay. Now, it's not always going to go into the nucleus. Sometimes it will just stay in the cytoplasm. Okay. And then it'll just change some proteins that are already there. So these pathways are really complicated. Like if you look back here, I mean, look how many steps are in the transduction phase alone. That's a lot of steps. This is all transduction. After this reception, this is all transduction until you get to right around here where you have an active protein. Okay, so that's a lot of transduction especially. And so these can get really messed up because there's a lot of steps. And if you mess up a single step, it's going to mess up the whole thing. So some examples of those things getting messed up are actually in diabetes. Cholera can do this to you. We're going to talk a little bit about that. Autoimmune diseases, sometimes that can result from the cell not being able to do its transduction pathways properly. And also cancer and some neurotoxins. Okay, so cholera is a disease that you can get by drinking contaminated water, and it's specifically typically going to be contaminated with human feces. So you're going to see this a lot in third world countries that don't quite have good water filtration systems or even good plumbing systems. Okay. And so what will happen is basically a bacteria, I don't need you to know the name, will colonize your small intestine and it'll start making a toxin. That toxin will mess with that G protein. Okay, so it's just going to mess with one of those plasma membrane proteins that is responsible for one of those transduction pathways. And this G protein is responsible for regulating salt and water secretion. And so it's basically going to make the G protein always active. And that's going to cause your intestinal cells to just start letting salts and water out all the time. And so this is going to cause some really, really awful diarrhea, and it can eventually kill the individual just because you lose so much water and so much salt. Even though you're drinking a lot of water, you're just losing it. Okay? Viagra, actually, funnily enough, is also going to involve signal transduction pathways. So with erectile dysfunction, sometimes a signal transduction pathway is to blame. And so this medicine will actually inhibit the hydrolysis of CGMP. So that was one of the early examples way back. And it will actually allow the signal to kind of relax the smooth muscle, and it'll affect your blood pressure, okay, and your blood flow. Okay. Now, here's the last point I really want to talk about is apoptosis. This is cell suicide or programmed cell death. If a cell undergoes apoptosis, it gets dismantled and digested. We really just break down those cells. And so basically, signals will get sent to the cell that will say, hey, you need to go. I'm sorry, but for the benefit of the multicellular organism, you need to go. And so it's going to activate a response just because it's cell communication, right? It receives a signal, the signal undergoes transduction, and then it will respond by killing itself. Okay. Now, why would a cell ever want to do this, or why would that help an organism? Well, sometimes it will help protect a neighboring cell from damage. Sometimes it's necessary for your development or your maintenance as your body is changing. And sometimes when this happens when it shouldn't, you actually can cause a lot of diseases. So like Parkinson's and Alzheimer's. And if you don't undergo apoptosis when you should, so if your cells are staying alive when they really should be dying, some of them, because some of your cells should be dying every day, right? That should happen. Then what you might get is some cells that are cancerous. And so then what they're going to do is they're going to reproduce like crazy, even though they should be killing themselves. Instead, they're reproducing themselves. So it's the exact opposite of what we want. And so that will eventually result in a cell creating 2 cells, which creates 4 cells, which creates 8, then 16, then 32, then 64, etc., until you eventually get a big tumor. Okay. So we see this pretty much throughout all cell types in the human body. There are some exceptions, but I don't want to worry about that. So on the left here, I have a normal white blood cell. So white blood cells are responsible for helping fight infections, right? And on the right, we have a white blood cell that is now undergoing apoptosis. So it's like in the middle of killing itself. And so you can just tell there's something wrong here, right? It looks like popcorn almost. So it's just kind of bubbling up in different areas, and we call those lobes, and it's shrinking in other areas. So it doesn't exactly look fun, right? But this is a normal part of cells in a multicellular organism. And so sometimes this will be, as I said, necessary for growth and development. So here I have the early embryo tissue, okay, of a mouse. And so this is the tissue of the hand or the paw. And what's going to happen is you can kind of see it's wider, more solid almost, right here, here, and here. And then there's like darker areas in the middle, like where it says interdigital tissue. So that means between digits, so between fingers. And so what's going to happen is between where the fingers should go, those cells are actually going to undergo apoptosis. They're going to kill themselves so that you get the proper shape. And so now this embryo is going to develop properly shaped paws. Okay, so that's going to do it for cell signaling. I know that was a lot of information, but I don't want you to get bogged down in every little detail. This is really what I need you to understand, is that there are 3 steps to cell communication. And the cellular response can happen in the nucleus, or it can happen in the cytoplasm. If it's happening in the nucleus, it's going to change your gene expression. Okay, we call that gene regulation. And if it's happening in the cytoplasm, it's probably just going to affect some kind of an enzyme or a protein inside of the cell. Okay, and remember that the response could be a cell committing apoptosis if necessary. So with that is going to do it for cell communication. And I will see you next time for chapter 12.